program. It's going to be another rather long session. Dr. Ramona Haebler is going to present material over the next 3 hours. And she will give us breaks in that time. Dr. Haebler did her veterinary training at Michigan State University and then stayed on there to be a graduate student to complete a PhD under Dr. Robert Leader. She tells me that she had a grant from the Dow Chemical Company and went to NIHS in North Carolina as a graduate student to conduct her research in the general field of environmental toxicology. Now, no doubt she can correct all that, give some fine detail to all the generalizations I just mentioned to you. She has been with the Environmental Protection Agency, working in the environmental lab at Narragansett, Rhode Island for the past 10 years. And so she's going to tell us something about the work that she's been doing.
Thank you. Thank you very much. It's really an honor to be here this afternoon and hopefully be able to share with you some of the knowledge I have, and especially some of the excitement I have about working with the species of marine mammals and other endangered species in the marine environment. I'll be mentioning a little bit about some sea turtles, which I think kind of got left out of our planning here, and then a little bit about marine birds. I'd like to, before I begin, describe how I plan to kind of break the talk up. I'm scheduled to have time available for the next 3 hours, and I'd like to separate that into basically 3 different segments. As I'm sure you're all aware that tomorrow morning Tom Lipscomb will also be speaking with you about marine mammals. So Tom and I have talked this over and in trying to coordinate things as best we can. The way we've divided it up is that I'll be speaking for the first hour this morning about marine mammals and their interface with pollution in the marine environment. Tomorrow morning, Tom will be speaking to you for about an hour on the infectious diseases of marine mammals. So hopefully, between the two of us, we'll cover both the infectious diseases as well as the environmentally related type diseases, or at least what we know about environmentally related risks to the animals. Sometimes there's not an awful lot that we really do know. In the second hour today, I'll be talking about the Exxon Valdez oil spill. You've already heard bits and pieces about it. And what I'd like to do is to present to you kind of a beginning to the end overview of how we approached doing that investigation and assessing the impacts of that major environmental disaster on wildlife. Tomorrow morning, Tom will be speaking with you for an hour very specifically on the impacts of that oil spill on sea otters, and I think particularly on the histopathological change associated with that exposure. So hopefully these, the way we've divvied it up will make a little bit of sense. And then in the third hour today, I'd like to talk about diseases of marine birds, and there I'll focus on both infectious diseases and some of the marine, or some of the pollution-related diseases. Much of this has already been covered, so again, we'll hopefully be able to coordinate things a little bit. So if I could have the lights down, we'll begin. As you've just heard, I've been working with the Environmental Protection Agency for the last 10 years, and our laboratory in Narragansett, Rhode Island focuses strictly on marine pollution. Although most of the other people at the laboratory work in the other aspects of marine pollution, I in particular and a few of my colleagues work on the effects of pollutants in the marine environment on marine mammals and other endangered species. So, just as a forewarning, I'm going to be coming at the talk today primarily from the perspective of the environment and pollution in the marine environment. So, we've been talking this morning and the last few days a lot about land animals and animals in the mountains and animals on the East Coast, and today I'd like to go heading right back out into the ocean and give you a little bit of background on the ocean to begin with. The oceans are a very complex ecosystem that provide critical habitat to literally thousands of species of both plants and animals. But these oceans aren't invincible, even though they may appear so. Man uses these waters, and in doing so, he has contaminated them through 3 basic mechanisms. Accidentally, through acute catastrophic events that cause a very sudden change in the health of a marine ecosystem. Intentionally, from the chronic discharge of human, animal, and chemical waste. And it's important for everyone to remember, I think, that when contamination goes into a stream or a drainage ditch, it eventually goes to another waterway, to another waterway, and to another waterway, which eventually ends up in the ocean as well. The third major category of marine pollution is persistent marine debris. This type of pollution has two major components. The first is intentional polluting from littering, from jerks and others, and also accidental pollution from lost fishing gear and the variety of other things that go over the sides of a ship in transportation. Each of these types of marine pollution may adversely affect the health and the well-being of marine mammals very severely. So what I'd like to do this afternoon is to first discuss a few fundamental principles about pollution and where it comes from, then describe the differential susceptibility of various pollutants to the specific kinds of marine mammals, and then finally discuss some case studies to give you an idea of not only how these types of pollution affect the animals, but how very difficult it is to really assess the problems that are caused in the animal, and then hopefully to be able to mitigate the problem. Though I don't agree with this statement, or I don't like it, and I doubt that any of us do, it is in fact the truth. And that is that the marine waters of the United States, and that includes the estuaries, the coastal waters, and the open oceans, are all used for the disposal of various types of waste. And it's all legal. Three major sources of pollutants enter the marine environment. These include point sources, which discharge municipal and chemical waste through effluent pipes, non-point sources, which refer to agricultural and urban runoff, and ocean dumping is the practice where wastes are transported by ship or barge to various designated marine sites and then discharged overboard. From these different sources, various pollutants are introduced into the marine environment. Typically, I think we often mistakenly think of marine pollution as being just chemicals, and that's not the case at all. Nutrients, pathogens, toxic chemicals, and metals come from both point and non-point sources, and the sewage sludge, the industrial waste, and the dredge material that is dumped at sea contains a very wide variety of contaminants. Marine mammals are the subset of true mammals that have evolved unique characteristics for survival in an aquatic environment. Since these groups listed here did not develop from a common phylogenetic line, they vary greatly in their anatomy, physiology, biochemistry, and habitat requirements. And it's because of these differences, the major groups of the marine mammals are at very different levels of risk from exposure to contaminants in the marine environment. And just as a side comment here, members of all of these groups of animals, with the one exception of the dugong, live in U.S. coastal waters. There are 6 species of sea turtles that live in U.S. coastal waters, and all of them are either on the endangered or threatened list. Unlike marine mammals, all sea turtles are really relatively rather similar, so they don't differ so dramatically in their risk associated with exposure to marine pollution. What I'd like to do now is to briefly run through these major categories of animals and discuss how they have a differential susceptibility to the major kinds of pollution. The animals I'd like to start with are the Cetacea. The order Cetacea is comprised of 2 suborders, mysticetes, such as shown here, which include 11 species of baleen whales, and the odontocetes, which are the toothed whales and dolphins, and there are 68 species of those. Many species of both mysticetes and odontocetes inhabit our U.S. coastal waters throughout the entire coastline. These suborders are differentiated according to the anatomical characteristics of their mouth and their feeding structures. And in fact, it's these different characteristics of their mouth anatomy and their feeding behavior that put them at differential risk to oil in the marine environment. Physical exposure to oil poses risk to cetaceans, especially to the baleen animals. This is a right whale skim feeding at the surface. Fouling of the baleen plates with oil may decrease the filtration capability of this animal. As you can imagine, there's not much space in between these baleen plates, and if this animal were going through an oiled area, whether it be a thin sheen or a thick moussy area, it would certainly foul up that baleen. Species at greatest risk are those that feed at the sea surface, such as the skim-feeding right whale, as well as humpbacks who feed at the sea surface like this, or who come crashing through the sea surface during lunge feeding behavior. Also, the bottom feeders, like the gray whale, are at risk from this type of behavior, and I'll mention those again in a few moments. Also at risk from exposure to oil, the thick weathered oil may also clog the breathing passages of the blowholes. There's conflicting evidence about whether cetaceans can detect and avoid oil slicks, which would clearly affect their exposure risk. Experimental studies of captive trained dolphins indicate they avoided oil slicks through either detection by visual, tactile, or echolocation means. However, in the field, observations describe cetaceans both swimming through and feeding in areas contaminated with oil. As far as the physical contact route of exposure, impairment of the thermoregulatory mechanism that is a serious problem for other types of marine mammals is really not thought to be a major risk for cetaceans for two primary reasons. One is that these animals tend to depend primarily on a thick blubber layer for insulation, and secondly, oil doesn't readily adhere to their smooth skin. Exposure to cetaceans to pollutants in the marine environment is thought to be almost exclusively through the dietary route. Within the cetaceans, between the baleen animals and the odontocetes, the diet of these animals varies tremendously. In the right whale that we saw skim feeding at the sea surface, this is what he was looking for. This giantest of one of the giantest of all whales eats strictly zooplankton. This hair-like material, for those of you who aren't familiar with baleen animals, the baleen plates have these long hair-like structures trailing on the back end of it, and this is an accumulation of this animal's dinner. So as you can imagine, they need to find very dense patches of zooplankton to be able to maintain that big animal. So anyways, their dietary prey sources vary from just zooplankton up to, this is the dietary sources for a humpback whale, ranging from the small zooplankton all the way up to fish and squid. Other animals at the very top of the food chain, such as the sperm whale, not only eat large fish, but they also eat many other marine mammals. So as you can imagine, these animals who are eating higher in the food chain have a greater chance of ingesting animals who have either bioaccumulated or biomagnified toxic chemicals. Very little is really known about the exposure or the effects of pollution in these animals. It's generally thought that the risk would be greatest in animals that either feed in nearshore waters, especially near industrialized areas or urbanized areas, or animals that feed high in the food chain on prey species which may have accumulated toxins. We're currently doing some work in our laboratory now looking at biopsy samples taken from free-ranging humpback whales out in the Gulf of Maine and other feeding areas up in the north, so to try to get some kind of a handle on the levels of chemical residues in the blubber of these animals, in animals that aren't dead. Unfortunately, and it's something we'll talk about a little bit later, one of the problems we have is usually the only animal that you can work with is an animal that has died from some unknown reason. So to be able to determine what is normal in an animal is very, very difficult. Two other scenarios of risk from contaminants in the marine environment to our great whales involve the gray whale and the right whale. Concern for the gray whale centers around their feeding habit of literally scooping large amounts of bottom sediment, which is then filtered through these baleen plates, and then they eat the benthic infauna. In the process, they're likely to ingest a significant amount of sediment, which may be highly contaminated. Concern has been especially intense within the last few years when there's been an increased mortality of gray whales up in Puget Sound. This just boggles my mind. I've also seen pictures taken of the bottom sediment with just these big gouges. I find it very humorous to think of these great big guys going down there with their mouths open and just scooping up all the stuff and then the mud flowing out the sides of their mouth. Whoops. Another whale where there is a special concern, especially in the present right now, is the right whale. And there's concern that nutrient loadings, and again, now we're getting away from talking strictly chemicals or oil. The concern here is that nutrient loadings may trigger toxic blooms, which could be especially damaging to the right whale, the most endangered of all whales in the world. This animal feeds in the very nearshore waters in the Gulf of Maine, and this issue of nutrient loadings is central to the endangered species consultation that is currently underway in order to permit the new outfall for the, it's called the MWRA, the Massachusetts Water Regulatory Authority. It's built a pipe, instead of dumping all of their sewage basically all over the edge of the sidewalk into Boston Harbor, where they've made really quite a mess. Instead, now they've built a pipe that goes 9 miles out into Boston Harbor to run all of this stuff out. But unfortunately, the pipe ends here in Stellwagen Bank, which is a National Marine Sanctuary and a primary feeding ground for both humpbacks and right whales and fin whales. It starts right about here. So there's a lot of controversy about this right now. To move on to seals now, there are 34, or pinnipeds, excuse me. There are 34 species of pinnipeds, and these can be divided into 3 major categories: the hair seals, the fur seals and the sea lions, and the walruses. Pinnipeds inhabit U.S. coastal waters along the entire west coast, and the east coast as far south as New Jersey and sometimes even further south. There are no pinnipeds in the Gulf of Mexico. 2 characteristics which vary among these groups can make their relative risk to adverse effects from oil somewhat different, and those are variations in their pelage and in their feeding habits, and I'll come back to those. Otherwise, within the pinnipeds, risks from exposure to oil are fairly consistent across the species, varying primarily with geographic distribution. Physical contact is a major route of exposure for pinnipeds when they run into oil in the environment. Pinnipeds have exceptionally large, protruding eyes, which make them very susceptible to toxic volatile compounds. Irritation of the eyes and the mucous membranes is reported following both experimental exposure to oil slicks as well as oil spills in the natural environment. Since these volatile compounds evaporate rapidly, usually within 24 to 48 hours, this risk should decrease with time following a single release. However, hazards associated with this mechanism of toxicity will be more severe during a well blowout or some other event where oil is discharged over an extended period of time. This is the northern fur seals. They're very dense up on Pribilof Islands where this photo was taken. The thick pelage of the northern fur seal provides insulation necessary to maintain thermoregulation, and in this way the northern fur seal is somewhat different than most other pinnipeds. Contamination with petroleum hydrocarbons removes natural oils and increases the heat transfer, thus causing a higher metabolic demand. Pinnipeds other than fur seals depend primarily on their thick blubber layer for insulation, and therefore are at much lower risk to thermal effects associated with fouling of the pelage. One exception, however, may be newborn phosid seals who may have to depend on their lanugo for a short while after birth. The lanugo is a thick prenatal fur coat, but sometimes is still present following birth. Unlike most marine mammals, the pinnipeds give birth on land, where the newborns then spend several days while moms are out foraging. The rookeries where these animals are born are usually rocky outcroppings, which, if they happen to be in the middle of an area where there is an oil spill, can entrap weathered oil and increase the risk of exposure. I'll be showing you some examples of that in a few moments. Though pinnipeds coated in oil can usually rid themselves of contamination within a short time, this is in contrast to the sea otter, it's thought that seals can usually rid themselves of the oil within a period of days to weeks. It has been suggested by some, though I don't think proven, but at least suggested that heavy oil may impair the swimming ability of these animals, even leading to the point of drowning. Considering inhalation as a route of exposure in the pinniped to oil in the environment, when this was done experimentally with ringed seals in Canada, the tissue residues of petroleum hydrocarbons were attributed to this inhalation route of exposure. There was damage to both the liver and kidneys in these experimental animals. In the field, a very common pathological finding in wild populations of pinnipeds is parasitism due to lungworms, and then of course they have all the associated secondary disease processes such as pneumonia that go along with disease entities such as this. As you can imagine, exposure to an inhalation toxicant or irritant may be especially damaging and dangerous to the individual animal who has pre-existing pulmonary disease. So again, I think it's important to keep in mind that when you read in the literature that inhalation exposure in pinnipeds may not be a real big deal, it's a deal, but it may not be a real big deal. I think it's important to remember whether those are generalizations or whether you're looking at individuals in the populations. If it's a bunch of harbor seals and you've got 1 million of them, it's no big deal, unfortunately. If you're dealing with endangered species, I think these kinds of thinking are very important. Experimental studies on ring seals using oral doses of radiolabeled petroleum hydrocarbons show that these compounds are absorbed into the bloodstream and distributed to various body tissues. The concentrations are highest in blubber and liver. Both the renal and the hepatic biliary enzyme systems appear to be effective in metabolizing these petroleum hydrocarbons to polar metabolites for excretion, primarily through induction of the mixed function oxidase system. The enzyme aryl hydrocarbon hydroxylase, identified in both liver and kidney tissue of ring seals, could be induced in vitro by exposure to crude oil. In gray seals and harbor seals, the hepatic MFO system is more developed in adults than it is in the young, which shouldn't be surprising. But this suggests that petroleum exposure may be more harmful to the newborn animals due to their metabolic incompetence. Tissue residues of petroleum hydrocarbons are present in lipid-rich tissues of several species of pinnipeds, indicating that these compounds are not completely eliminated from the body and therefore may pose a long-term risk to the animal. This risk is most likely highest when adipose stores deplete during migration, molding, molting, or lactation. Transfer of these compounds through the very lipid-rich milk may further endanger neonates. I was amazed when I started working with marine mammals, and especially, I was especially interested in lactational transfer, to learn that the fat content of these animals is frequently up in the range of 40 to 45 up to 55% milk fat. It's a lot. Both seals and sea lions feed primarily on fish, which live in the water column, and therefore risk from petroleum hydrocarbons through the food web is really thought to be minimal. However, the walruses may be at greater risk since they feed on benthic infauna, organisms known to bioaccumulate PAHs. Ingestion of oil during grooming activity does not appear to be a major source of exposure in pinnipeds, as it is in other species such as the sea otter and the polar bear, which we'll discuss in a few moments. Pinnipeds are most likely exposed to anthropogenic contaminants through the dietary route, since they feed high in the food web on prey species that are known to bioaccumulate anthropogenic chemicals. In the world of marine mammals and pollution, I think the literature is by far the most complete in this area of contaminants and seals. And I would encourage any of you who are particularly interested in this to turn to the literature. I think some of the very nicest work has been done. And in fact, as you all know, here in this country, we have the Marine Mammal Protection Act, which absolutely eliminates any possible controlled exposure type studies. But Peter Reinders from Holland has done a very, very nice study where he's actually shown causal link between feeding fish from contaminated waters and therefore having lots of PCB in them, and feeding fish from cleaner waters to harbor seals and actually causing reproductive suppression. And he's now working on working through the biochemical and hormonal mechanisms of that. So there really is some very good literature out there. I'd like to move now to manatees, who are some of my very, very favorite animals. The order Sirenia contains both the manatees and the dugongs, but as I mentioned, only the manatee lives in our U.S. coastal waters. These are extremely endangered animals. There's only somewhere around 1,000 to 1,200 of them that still live in the U.S. Population, and I believe that our population is about the largest in the world of the West Indian manatee. These animals live in the warm coastal waters of southern Florida and up to the north, and occasionally you'll see sightings in the adjoining states, but not too often. Like Cetacea, these animals are totally aquatic, but unlike any other marine mammal species, manatees are totally herbivorous, feeding on vegetation in the shallow coastal waters and the riverine systems. Manatees are intolerant, very intolerant to cold temperatures, and they depend on warm water to maintain their thermal regulation. There's very little information available from the literature about the potential effects of oil on these particular animals. There's never been a major oil spill in Florida, but there has been a lot of oil spilled into the Persian Gulf, as we all know. During the Persian Gulf War, the Iran-Iraq War in 1983, and the Persian Gulf War in 1991, millions and millions and millions of gallons of oil were spilled. And during both of those events, from flyovers and all, carcasses of dugongs were identified, as well as the carcasses of many other species of animals. But because of the war conditions, no one could get into the field to examine these animals until they were very, very autolyzed. So unfortunately, the exact effects of oil on these animals remains unknown. But I think that by understanding the manatees' natural habitat requirements and something about their natural history, I think we can speculate on what the detrimental effects on the manatees would be if, in fact, there would be an oil spill in Florida. And I think this is an important kind of thinking to do ahead of time, because I think sooner or later it is going to happen. If oil were spilled in a manatee habitat, I think they would likely contact surface oil, which may irritate their eyes, their mucous membranes, or clog their nostrils. Though direct thermoregulatory effects probably would not be terribly significant, if the manatees were driven out of their warm water habitat back out into colder waters, I think that they may actually succumb to hypothermia. And also, if this oil were to wash into their areas where they eat and contaminate their shallow water vegetation, I think there would be a good possibility of ingestion. As I mentioned a moment ago, manatees are totally herbivorous, eating only seagrasses. So logically, from the whole business of animals who eat low in the food chain, where there would be minimal bioaccumulation or biomagnification, they would have minimal exposure. So we've talked about right whales being pretty much towards the bottom and sperm whales up here at the top. Well, manatees would be down here even lower than animals who eat zooplankton. But it is a little bit more risky than that. One is that there has been some work done, although they haven't found too much, on looking at herbicide accumulation in plants that these animals eat. That work has been done, most of it done by the Fish and Wildlife Service in Florida. It doesn't seem to pose a big risk. I think one of the interesting things of these animals that I think may be of more risk to the animal is the fact that it appears that they like to drink fresh water. And the sources of the fresh water that they drink from can be either sometimes the pipes of sewage treatment plants or the effluent pipes from the pulp and paper mills that are in their habitat up in northern Florida. And in fact, there have been a few, and I don't know what the number is now, but it's like 2 or 3 or 4, I think, cases of toxoplasmosis that have been documented at the University of Florida. And although you can't be sure, the theory, the working theory with the folks at Fish and Wildlife Service who I know, feel that it's probably from the kitty litter going down the toilet through the sewage treatment plant and out the effluent pipe and contaminating the water where the manatees drink. Just to note, an unusual anatomical variation of these animals, their mammary glands are in the axillary area, so you may see things like this. But I wanted to just leave the discussion on the manatees with the comment that these are extremely endangered animals. And I think that when there is any possibility to save one animal, we're not talking about populations here, if we have a disaster anywhere near the magnitude of what happened in Alaska or the dolphin die-off a few years ago, I think we will lose this species forever. Sea otters are the only marine mammal within the family Mustelidae. They are found along the west coast of the United States, especially in Southern California, with occasional sightings as far north as Washington. Another population of sea otters occurs in the Gulf of Alaska and all the way out the Aleutian chain, but this northern population is not listed as either threatened or endangered. As you've all heard before today, you've heard today some, you'll hear a lot more yet today and tomorrow, these little guys are very susceptible to exposure to oil in the environment. Sea otters are the smallest of all marine mammals, and they only have about 3% of body fat. To maintain their body temperature, they depend on this very elegant fur coat that they have, and they also depend highly on their metabolic rate, which is about 2.5 times what we find in most other terrestrial mammals. When oil destroys the integrity of this fur, the insulating layer is lost, and the otters become hypothermic and can die very, very quickly. If they don't die from hypothermia and they're lightly oiled, they are manic little groomers. They have to look spectacular at all times, and they will lick and groom until they do get the oil off, but in the process they will ingest whatever oil is present on their fur. I'll talk talk more about this during our discussion of the Exxon Valdez incident as a whole, but prior to my going to Valdez, I really thought that oil was kind of like what you put in your car. And I had no concept of what a devastating compound this can be. This was not at all uncommon of what we found. This is a baby sea otter that came in dead. So there's not much question. It doesn't take a rocket scientist to figure out why this little guy didn't make it. Sea otters feed primarily on macroinvertebrates, prey species that are known to bioaccumulate petroleum hydrocarbon residues. Thus, sea otters are at risk also to ingestion from oil through not only their grooming behavior, but also through the ingestion of contaminated food. Sea otters do not appear to consistently either detect or avoid oil-covered waters, either in the field or during experimental exposures. Exposure of the sea otter to anthropogenic chemicals also comes primarily through prey species that are known to bioaccumulate anthropogenic toxins. Thus far, from what I know in the literature, there is not much to indicate that they seem to be bioaccumulating very much, though, which is great news for once. Maybe I just haven't read the literature enough, too. Unfortunately, the polar bears are kind of going off the top of the screen there. But the polar bear is the only marine species in the family Ursidae. These Arctic inhabitants are thought to have evolved from the Siberian brown bear. Polar bears are classified as marine mammals, although some people will argue this point. But the people who put them in the category of marine mammals say that they do belong there since they are dependent on the marine ecosystem for their survival, particularly their food source. They live on pack ice and they spend a considerable amount of time in the sea. They're tremendous swimmers in search of their prey. One of their main prey species are seals. They are considered to be at risk from oil in the environment, both through oil coming from spills as well as blowouts associated with oil and gas exploration in the Arctic. They have never, fortunately, been exposed to a serious accidental spill. They obviously were not down in the area of the Exxon Valdez. But there have been some experimental exposures done by the Canadian government that were done. Unfortunately, they lost some of the animals, but we learned a tremendous amount about these animals. They were exposed to oil in a tank and to a sheen and brought out, and they seemed to be doing just pretty dandy, and they were monitored very closely. They had a very thorough protocol. And with time, and I think if I remember correctly, it was quite a bit of time, like 2 weeks or so, these animals started to crash. And in fact, they died of primarily renal failure and also hepatic failure. So it was learned from that experiment that, in fact, these animals appear to be very sensitive to toxic effects from the oil. Just briefly to mention sea turtles. We have a lot of sea turtles, although they are decreasing in numbers. All 6 species of sea turtles are threatened or endangered, as I already mentioned. Sea turtles live, many in the Gulf of Mexico, up the east coast into the Gulf of Maine, and quite a bit on the west coast as well. Nesting beaches are found in the Texas and Florida area. Feeding behavior of the turtles is unusual. Up until a few years ago, it was thought, at least by the federal agencies, the one I work for anyways, that you didn't have to worry about these animals when it came to marine pollution because they just kind of crawled out of the sand and went to sea and lived this mysterious life and ate pelagically, and you know, chemicals didn't get out that far. But that's not the truth. It's found now that yes, in fact, that happens, and they go out there and they spend a few years out at sea feeding somewhere off in the Sargasso Sea or whatever, but then they return. And they return, the Kemp's Ridley, for example, returns at about 3 to 5 years of age to the nice warm water estuaries along the East Coast. They're the ones I know most about. And they come in to Long Island Sound, Pamlico Sound, those kinds of places, and feed on the bottom animals like crabs, animals who are known to bioaccumulate a lot of toxins. So they certainly are at risk to exposure to some of these. What the biological significance of it is, we don't know yet. We've recently just completed a study on contaminant levels in Kemp's Ridley turtles. Chemicals are certainly there. What the effects are, we'll have to keep working on that. I'd like to shift gears just a little bit now. I think we've talked about each of the different species and how they may or may not be more or less susceptible to different kinds of chemicals. And I'd like to talk about what chemicals do in the environment and how they're so unbelievably difficult to deal with and to understand how they may affect the animal. When I did my training, as Dr. Cordes mentioned, I was funded by Dow Chemical Corporation, and I spent a lot of time up in their toxicology unit. And then I did my actual research down in the lab of developmental and reproductive tox at NIEHS. And we had lots of rats, and you could dose them and not dose others, and it was really nice, and everything was controlled, and they all slept on the same thing, and it was marvelous. It was a little boring sometimes, but it was nice scientifically. Unfortunately, in the oceans, it's the total opposite extreme of that. So to try to make any good scientific sense out of what we see is very difficult, and I think we need to be very cautious about that. I'd like to make just a few comments about contaminants in the marine environment. They have really sunk into me in the last few years since I've been at our laboratory. Chronic pollution in the marine environment has absolutely no boundaries, no temporal boundaries, no spatial boundaries. What comes out the end of any given pipe today may be the same as it was yesterday, or it may be completely different. There aren't any fences in the ocean to prohibit the mixing of contaminants from all these various sources, and especially if you think about out here along the New Jersey, New York areas, there's just a multitude of point sources in addition to the non-point sources. Depending on the tides, the currents, and the atmospheric transport, pollutants can travel extremely long distances. The global ocean is contaminated literally from the Arctic to the Antarctic. Certain xenobiotics may enter the food web and bioconcentrate or biomagnify as they pass from one level to another. And the effects of certain environmental contaminants may also influence the ecosystem in very indirect ways, such as I mentioned a little bit earlier. It's possible that nutrient loading, such as phosphorus and nitrogens, may alter the media in which certain forms of algal blooms can preferentially proliferate, and then these blooms can become extremely toxic and very dangerous. Certain environmental factors may also interact or synergize with natural phenomena. For example, a change in water temperature can enhance bacterial or viral growth rates. A combination of stressors like this may play a role in the increased incidence of these natural disease processes observed in the marine system. I'd like to use a case study to try to use it as an example of some of all these complicated factors that are so very difficult to deal with. In recent years, there have been many unusual events of marine mammal morbidity or mortality in various parts of our country, as well as especially Europe. One of the most dramatic was the mass mortality of seals in the North Sea a few years ago, when over 17,000 seals died. Though now a specific organism has been identified through some very lovely work by a lot of very bright veterinarians and has now been characterized as a phocine distemper virus, there was and there still continues to be a lot of scientific concern that pollution may have played some type of a role in the onset of this disease process. In fact, many of these unusual events can be explained by natural phenomena, especially infectious organisms. However, though there is evidence that these natural phenomena may be the ultimate cause of death, they may not be the primary or the only etiology. I think there's a good possibility that a number of stressors may be acting together to bring a particular disease on. The case study I'd like to use as an example of how difficult it is to do a good disease investigation dealing with marine mammals is the dolphin mortality event that occurred here along the East Coast in 1987-88. The mass mortality of bottlenose dolphins along the East Coast in 87-88 is a case study illustrating the absolute complexity and frustrations of trying to find the cause of death in a vast number of animals. Prior to 1987, the prevailing attitude among most federal agencies, mine included, was that pollutants in the marine environment really didn't adversely affect marine mammals. It really wasn't even something to be concerned about. But then in July of 1987, the Smithsonian Institution's Marine Mammal Stranding Network alerted NOAA, the trustee agency, that unprecedented numbers of marine mammals, bottlenose dolphins in particular, were washing ashore in the lower Chesapeake Bay area and the Virginia Beach areas of the mid-Atlantic coast. These animals were either very ill or they were already dead when they came ashore. Between June of 1987 and March of 1988, 740 carcasses were recovered for examination. The number of carcasses retrieved is thought to represent only a very small percentage of the animals actually affected, since typically most carcasses are either blown out to sea or scavenged or sunk. Jerry Scott et al. In 1988 estimated that greater than 50% of the coastal migratory stock of bottlenose dolphins died during this event, and many more were left severely debilitated. Starting in June, the incidence of dolphin mortality started up here in the New Jersey area, if we can find it. New Jersey, New York, and literally started in June, and then as the months passed, it kept on going, and it reached Florida in December. There was tremendous public and scientific concern focused on this event. Many fears centered around the possibility that there might be either an environmental pollution problem causing this, or some type of an infectious disease process going on with human health implications. The bottlenose dolphin, a marine mammal, was the only animal involved. There were no fish, there were no crabs, there were no anything. So there was a lot of concern that this may be some type of a mammalian pathogen. That was also a big concern for the people working on the dolphins. A formal investigation was conducted under the authority of NOAA, the trustee agency, and EPA was asked to participate, although as a minor participator, in the dolphin response team. And I'd like to show you a little bit of what I was able to see and do while I was on site with these animals. The animals presented with a variety of clinical and gross lesions that couldn't be explained by any one etiology. There was evidence of viral infection. As you can see, there are vesicles along the side of the lips here, and they were also present inside the mouth. This was a live capture operation where animals were more or less selected at random, and they were netted in and brought on board for examination. So these appeared to be, grossly, they appeared to be early viral lesions, vesicles. The animals otherwise did not appear particularly ill. When animals died, and we were able to examine them then, this is the type of lesion that we saw inside the mouths of these animals. And the interpretation was that this was probably the late stage of what we had seen in the live animals, probably where a vesicle developed, it broke, and had secondary bacterial infection. There was evidence definitely of viral infection in these animals, and at the end of the study, there was thought to be 2 or maybe 3 viruses present. Originally, it was thought that this was probably a viral etiology for this disease process, but in fact, of the 2 or 3 viruses that were found, none of them were determined to be pathogenic. There was no, they were common viruses, they were typically non-pathogenic, and there was no pattern. One animal had one and one had another, but there was no common ground. In most animals, there was evidence of bacterial infection, usually quite severe bacterial infection, as seen here in this pneumonic lung, as well as in this abdomen that has chronic liver disease, peritonitis, and extensive adhesions. Though bacterial infection appeared to play a very important role in the disease processes of these dolphins, still no one organism or group of organisms could be identified as the true etiology of this disease. 22 different species of bacteria were identified and isolated. Most of these were organisms that were indigenous to the waters that the animals were found in. Most of them were non-pathogenic. Many of them were Vibrio and other common organisms in the marine environment. Since there didn't appear to be a single infectious organism that was causing this devastating disease, attention was then focused on the possibility of a biotoxin as the likely causative agent. And Dr. Gerasi, who was the principal investigator on this study, in his final report, he focuses on the likelihood, although not a definitive likelihood, but the likelihood that Tychodiscus brevis, Florida's red tide toxin, was probably the etiologic agent in this disease process that caused suppression of the immune system, leading these animals to become more susceptible to indigenous organisms. Unfortunately, or fortunately, I don't know which, the scientific process is never very clean-cut, and this was the final line of the final report that came out of the government. But it was met with a lot of controversy within the scientific community, and most of the controversy centered around the fact that these animals had high levels of contaminants, and we really didn't know what that meant. As a result, our laboratory, along with the Smithsonian Institution, did a study on a subset of these animals to try to look more in depth at what kinds of contaminants were there and in what levels. So we were able to do a sample of about 20 animals, I believe it was. And we had two objectives in doing this study. First was we wanted to look for contaminants that we knew were going to be in the water. We know in the ocean and in the sediment there are, A, B, and C. There's DDT and there's PCB and all that. We wanted to know what levels of those compounds were present in these animals. And of all the compounds that we looked for, we found almost all of them. And this is a representative of the types of results that we found. But this also points out one of the biggest problems that we have in working with marine mammals and trying to understand the effects of these pollutants on them, and that is that we have no way of interpreting this data. We don't know how this compares to other animals, to other dolphins or other marine mammals, because there's very little baseline data out there. And we don't know, because it doesn't exist yet, the type of research we need to do to determine the biological significance of this. So the best we could do is to compare our numbers to numbers from animals reported in the literature. Unfortunately, you always have some methodological differences, but I'll be showing you the next slide a comparison of our numbers here of the PCBs that were found in these animals. This is a comparison of our animals over here on the right, PCB levels in males and females, compared to gray seals from Sweden, white-beaked dolphins from Canada, Dall's porpoise from Japan, and fish from the Great Lakes. As you can see, what we can say from this is that the level of PCB in the blubber of the animals that we evaluated was very significantly higher than that found in marine mammals from other parts of the world, as well as from Great Lakes fish. But again, the biological significance of that we can't comment on at this point. Our second objective in this study was to try to look for, or ask the question anyways, is there something out there that we don't know is out there? Basically, so a lot of times we're not smart enough to know what question to ask, and we're pretty sure we weren't smart enough to know what all chemicals were out there. So the chemist I was working with is a fantastic analytical chemist and quite a detective too, I think. And so he basically did the kinds of work to see what else was out there. And much to our surprise, we found that these animals had quite a lot of polybrominated chemicals in their tissues. This is a class of chemicals that not only did we not know were present in animals, but we didn't even know they were present as a contaminant in U.S. coastal waters. Whoops. We're losing things here. Hold on. I'll tell you, I'm going to be in a hell of a mess later. I've got to be able to keep my right hand straight from my left hand. I can't even tell front from back here. Okay, we found polybrominated biphenyls, and we also found polybrominated diphenyl ethers. These compounds are flame retardants. Like I said, we did not know that they were contaminants in the coastal waters. There have been reports from Sweden that they were contaminants over there, but this was the first time we were aware of it here. We also, unfortunately, have a number of these brominated compounds that we haven't yet been able to identify, and we're still working on those. We feel that the findings of this study are significant. I don't think we found the cause of the disease or why they died, but I think it's very important to look at the levels of the contaminants that we have there, to look for chemicals that we don't expect to find there, and to use this information and integrate it into the more comprehensive and overall study set altogether. Unfortunately, the cause of this mass mortality may always remain a mystery. I think, though, that Tom Lipscomb, I hope he talks about it, I think he has some new and very exciting news. But I think some very good things came out of this event. Prior to this time, it was very difficult to work with marine mammals. There was a lot of personalities and jealousies and things like that. And when all this happened, not only did a lot of normal people get upset, but even the Congress got upset. And as a result of all this, an amendment was made to the Marine Mammal Protection Act that calls for a number of different actions. One of them is a contingency plan that will put into place actions that should occur if and when these types of mortalities happen again. An unusual marine mammal mortality working group has been formed to advise the Secretary of Commerce and the Secretary of the Interior in light of unusual mortalities like that, and to advise them on how to conduct the scientific investigation. And both Tom and I sit on those groups. A marine mammal tissue bank has been formed where tissues from incidentally taken animals will be archived for the future, so that if something like this happens again, we'll be able to go in and drag out tissue to basically use as a baseline. A number of different activities have occurred, and so hopefully they will help to lead us onto being able to protect these animals as well as to protect their habitat. And like I said, I hope that as part of this whole brouhaha that went on, the slides, the histological slides from this die-off were returned to the Armed Forces Institute of Pathology, where they will be archived for future use. And this will be done in all events, I believe. But anyways, Tom and some others at AFIP have been working on those slides, and they found some new and very exciting findings. I'd like to spend just the last few minutes of this first hour talking about the third kind of pollution, the persistent marine debris. And though this isn't rocket science, you know, in that we're not cloning extinct species or anything like that, it's very important, it's very, very deadly, and it's killing off an awful lot of our marine mammals, our protected animals, and especially our endangered species. Persistent marine debris harms animals in two main ways: by entanglement excuse me, and by ingestion. Entanglement can cause death due to strangulation, suffocation, or exhaustion. And ingested plastic or other debris may cause blockages or ruptures of the digestive tract. Pinnipeds are one of the best studied species, especially the northern fur seal. The most common mechanism of harm in these animals is entanglement in fishing nets or lines. It seems that pinnipeds have this very natural curiosity that we see in them a lot, to stick their nose in something, but unfortunately with marine debris it can often be very, very deadly. The effects on pinnipeds are reported worldwide in many, in fact probably almost all species of pinnipeds, including the endangered monk seal. Whales and dolphins frequently fall prey to the hazards of marine debris. Many observers worldwide report whales and dolphins entangled in fishing nets. Scientists estimate up in my area of the world in the Northeast that 40 to 50% of the endangered right and humpback whales in the North Atlantic waters have entanglement scars. I mean, that's just incredible. I couldn't believe it when I first heard that, and then I talked to the scientists that did the work, and then I've been seeing more and more of the animals, and I believe it's really true. And I'd like to show you one example of how this can happen. And this is a very personal story for me because it happened within a quarter mile of where I live up in Rhode Island. This is a humpback whale that became entangled in lobster gear. You can see two lobster traps here tied together by ropes, and they set them out in lines. And this happened just off Hazard Rocks in Narragansett. It's a very happy story, though. For those of you who know Larry Dunn and the other folks at Mystic Aquarium, they have a whale rescue team, or I'm not exactly sure how they refer to themselves. In fact, there are several of these groups throughout the Northeast coast, and they're well prepared and well trained and all. But anyways, Larry and his group of cowboys were able to come in, and it's really very dangerous. There are big rocks, surf crashing in, nice lobster trap set right amongst the little rocks and everything. And this whale was flailing all around. But they came in a Zodiac and dive gear, and they were able to enter the water and cut the animal free with knives, cut the ropes, and swam off, and as far as they could tell, lived happily ever after. So it's nice to be able to tell a happy story once in a while. Unfortunately, our endangered manatees also can fall prey to ingested debris in particular. There have been several reports in the literature of ingestion of plastics that have caused blockages, sharp objects that have penetrated the gut, and the animals have died of peritonitis. Sea turtles are also frequent victims of both entanglement in lines like this, but also ingestion of plastics. This is a local sea turtle that washed up on First Beach over in report last summer that I had the opportunity to examine. And though we don't know why he died, the animal was in really terrific shape. I don't think there's anything to do with it. But he had a massive lesion here. And our theory from the way it looked like it had been an entanglement lesion with a rope wrap around it, all kinds of proliferative fibrous tissue. I thought maybe there was going to be some type of a neat cancer under there or something, but it wasn't. It was just lots of proliferative old scar tissue. He's a great big leatherback, and his skull is going to be in my office shortly. Excuse me. Birds, too. We'll talk more about birds later, but birds are at tremendous risk from marine pollution. One of the things that is a serious hazard to these guys is all these little plastic pellets floating around out there in the ocean, and mama bird eats these. So not only is it not good for the mama bird, who derives no nutrition from it, but also it can have devastating effect if she has chicks. As we all know, the way that mom feeds a baby for the first while frequently is by regurgitating partially digested food material. And so if what she is giving back to her baby is plastic instead of food, it will affect not only the mother, but the second generation as well. Also, fishing line is devastating. This is something that I think people just don't think about it. I think if they had any idea how dangerous this stuff was. It's all over down in front of our lab. But anyways, the animals pick up a little piece of fishing line. It cannot necessarily kill the animal directly, but if it becomes entangled, it can pick up more and more and more, and eventually they can get caught like this and die. And there's no reason for it. Entanglement's also a dangerous threat to fish. Fish are at risk most commonly from ghost nets and other common debris in the ocean, but they also can die from things like this. So to summarize for our first hour here, we've talked about some major categories of pollution and all the potentially severe adverse effects that they may have on the health of the animal. But I think it's important, and as we go along, and I seem to preach about the animals and our side of it, the side of the animals, I think it's very important to remember that we can't just stop all this activity of man in the ocean. Man needs the oceans, and we use them for some very critical needs that we have no other alternative for. So I think our goal, and it's important for me to keep this in mind constantly, our goal is in managing the oceans is to balance our needs so that hopefully we can, both the animal as well as man can benefit from them as much as possible. So thank you. Let's take about a 5 or 10 minute break, and we'll talk about Alaska next. Dr. Nielsen has gone over many of these things just an hour ago, but anyways, when I was trying to learn this kind of stuff, you hear it once and it kind of goes in for about 2 seconds, and you hear it twice, and maybe by the 4th or 5th time it will stay. But I think these are important concepts to remember, and that is that many species of marine mammals suffer adverse health effects following exposure to oil in the marine environment. But the level of risk associated with exposure to oil differs greatly from one species of marine mammal to another. Frequently forgotten facts about oil in the marine environment are that all oil spilled in the marine environment is not the same. Some of it's diesel fuel, some of it's crude. It can be anything in between. And the second thing is that once the oil goes into the water, it doesn't stay that way indefinitely. Oil changes with time. The primary routes of exposure to the animals are physical contact, inhalation, and ingestion. The severity of the risk from physical contact with oil varies depending on the texture of the body surface, the frequency and the duration of the exposure, and the type of oil. Toxicity of the oil, the physical and the chemical parameters of petroleum products determine the toxicity and the persistence in the marine environment. The acute toxicity correlates directly with the concentration of the light aromatic hydrocarbons, and the low molecular weight compounds volatilize at the air-sea interface where the marine mammals have to come to breathe. So again, this may be putting them at a higher risk to exposure and to adverse effects than other species in the marine environment. Heavy crude oils pose severe risk to both animals and the environment due to just their physical properties alone. Remember in the last talk I showed you that one little baby sea otter. That's thick stuff. It's very tenacious. And also with time, the oil becomes emulsified into what is called a mousse. This is a thick oil and water emulsion, which is extremely hazardous, not only to coating animals, but also to coating the beaches and the rookeries. So now I'd like to talk about the Valdez spill. This ship, the Exxon Valdez oil tanker, crashed into Bligh Reef on Good Friday of 1989, and it spilled over 13 million gallons of Prudhoe Bay crude oil into the environment. It was by far the worst marine environmental disaster that we've ever had, and hopefully that we will never have another one like this. Literally, overnight, it changed this unbelievably beautiful, pristine, and productive environment into a severely contaminated area. And that's the real key characteristics of these accidental episodic events. You don't have to have a contaminated area to start with. It can be beautiful. But literally, bam, and everything changes. When I was home on the East Coast in Rhode Island when all this happened, I saw maps like this, and this is where the tanker crashed, up here in Bligh Reef, just outside the mouth of Valdez Harbor. And then with time, they started making these cute little diagrams of how the oil spread. And it was very impressive. I went up to the spill about 7 to 10 days after the spill. This one is 3 weeks post-spill. This is very impressive. It was a huge spill. But I found that even when I was up there, until I saw this, I really fully didn't appreciate the magnitude of the spill. This is the same area overlain on the east coast of the United States, and that is if the Exxon Valdez crashed here, this spill would have gone all the way down to Cape Hatteras in North Carolina. Within 3 weeks, this spill area covered 9,600 square miles of ocean and over 1,000 miles of shoreline. When this happened, these long finger-like leads of oil flowed across the sea surface, and they often killed any and all wildlife in its path. A spill of this magnitude had absolutely massive effects. Oil, when it hit the beaches, Prince William Sound, for any of you who have not been there, is one of the most spectacular places on Earth. Oceans literally rise out of the ocean and go several thousand feet up. And the beaches are this real rocky, spectacular area. But it was literally covered with oil, slippery as hell. And pools of oil literally formed, and the oil went over a meter deep down into the beach material. As I said, I got up there about a week after the spill happened, and the job that I was assigned when I went up was to try to assess the impacts of oil, this oil spilled into the marine environment, on marine mammals in particular, wildlife in general. And I had the opportunity to work with scientists from several other agencies while I was there, and like I said, Tom Lipscomb will be speaking with you tomorrow morning. Very quickly after the spill, a massive effort was mounted by the private citizens, by the government agencies, and by Exxon and their contractors to go out into Prince William Sound and try to recover any and all animals possible so that we could have an opportunity to evaluate the impacts on these animals. It was a very devastating event, but I'll have to admit, from a pathologist's perspective, especially one who's interested in marine mammals, it was really the opportunity of a lifetime at the same time. So a lot of mixed, ambiguous feelings about this. The spill happened outside of Valdez, and then it went in that direction. So it was a very difficult thing to deal with. In that overlay that I showed you there of the East Coast, as you know, here on the East Coast, we have roads and cities and airports and boats and harbors and all that kind of stuff. You don't have that in Alaska. In order to get to the spill from Valdez, you either had to fly by float plane or you had to go out by a good-sized boat, and you had to pretty much stay out there. The weather up in Prince William Sound in March and April and May is very unpredictable, and it can be very dangerous, especially for flying. So anyways, a massive effort was undertaken to try to collect these animals and bring them in for examination. This is a very difficult job. That not only did they have bad weather, but Prince William Sound is a very big area. As I said, it covered 9,600 square miles of ocean, the oil did. And what they were looking for when they were looking for sea otters were little oiled footballs like this. You can see that the flippers are hanging down here under the water and the head is up there somewhere. So these were hard to find. So again, it goes back to the point that they did bring in around 1,000, but I think that was only a very small number of the animals that were found. They were also looking for birds, and as you can imagine, here too, it was very difficult to identify these black oiled carcasses on black oiled beaches. Even so, I think we did get, even though it was hard work, I know we missed a lot of animals, but we did get a very large number of carcasses brought in for examination, as well as live birds brought in for examination and hopefully for rehabilitation. I'd like to take just a few minutes and talk about the live animals, the animals that were brought in alive. Some of them were brought in alive, and we learned a great deal from them, and some were released. Some were brought in alive and they did not make it, and these animals then died, and these are the ones that were then necropsied, and those are the ones that Tom will be speaking to you about tomorrow. So I'd like to take a few minutes and describe how these animals, how and why these animals were brought in and how they were treated. The sea otter is a really fascinating little guy. He's cute. He's not as nice as he looks. He can be a little mean son of a bitch too. But anyways, the interesting thing about him in the world of marine mammals is that you basically can bring him on shore and treat him like a dog. Now, you can't put a whale in a cat carrier, but you can put a sea otter in a dog carrier and really get away with it for quite a period of time. So anyways, a major effort was mounted for a number of reasons: scientific, political, PR, who knows what. And people went out to try to gather up these animals and bring them in. One of the major ways that this was done was through capture nets or tangle nets like this. So people were living out in the Sound on boats or in little cabins and working out of zodiacs, and then they'd set lines like this where a net is suspended down into the water. Hopefully the sea otter would swim into it, get tangled up, and then they could bring him on board. When they brought the animal on board a boat, again, like I mentioned, you treat him much like a dog or like a coon or like a wild animal. In this particular picture, the animal is being weighed. The animal is brought on board, given an initial examination, assigned a number, all the routine things that you do for triage. Got more things going here. Then the animals were literally put in dog carriers, and they were held for transportation. We felt it was very important to move the animals off the boat and get them into the treatment center as soon as possible. So the animals were transported by either helicopter or seaplane back to the rehabilitation centers as quickly as possible. And that meant several times a day. There were 2 rehabilitation centers set up. The first one was done in Valdez immediately out of whatever was available. In the gymnasium of a high school, out of dog crates, out of anything they could get their hands on. They did the best they could. It didn't work out too well. There were lots of problems with air handling, with noise, with lights, and things like that. Within 3 weeks, the second rehabilitation center came online in Seward, Alaska. So Valdez is here, the oil's going like this, and so they set up a second one down here in Seward so the animals didn't have to be transferred so far. Within 3 weeks, they had made just an immense amount of progress in being able to design and construct a very nice facility. So a combination of some good rehab people and some good old Alaskan ingenuity, and they did this primarily out of ATCO trailers that were empty, and then whatever you could get from the local hardware store or fish supply store. So these were the wash tables. When the animals came in, the first thing that usually needed to be done was to wash them. Usually was about a 2-hour event, and the animals needed to be either anesthetized or at least tranquilized, and then scrubbed, and this is that very tenacious stuff, so it took a while. But anyways, you can see there's a nice table made out of fish netting here, drainage for the water, and then once the animals were washed and stabilized and out of intensive care, they went into outside cages like this. This is under construction still. This is the ATCO trailer in the back where the surgical facilities were, or the treatment facilities. This was done in like a courtyard setting, so the animals had a lot of privacy. These are the outside cages. You can see here they're made out of a fish tote, PVC piping, some fish netting, and then all of them, there's going to be another row here. All of these had flowed through seawater. Looking down into one of those cages, you can see the nice seawater pool here. The animals, as I mentioned earlier, have a tremendous desire to groom and to clean their fur coat. So this gave them an opportunity to get in the water and clean as much as possible. But also there were these haul-out areas here where these animals were very weak and quite ill. They could get out there and rest. As you can see, there are 2 animals in this cage. These are very social animals, and it made a tremendous improvement in their recovery to be able to be housed at least in pairs and then eventually in larger groups. Again, if there were more time, it's an interesting study to compare this type of cage with the earlier ones and how quickly people learned to improve for the care of these animals. As the animals then became more healthy and more stable and were able to swim a little and dive for their food, they were moved into these, what we called swimming pools. These pools were about 5 feet deep. Animals were housed in groups of maybe 4, 5, 6, 7. Mothers with babies were put in here. And now instead of being just kind of handed the food, frequently live food was thrown in and the animals could dive for it and swim for it. Food was a very important part of their diet. As I mentioned earlier, they had a very high metabolic rate, about 2.5 times most mammals, and they need to eat a lot, about 25% of their body weight per day. And they don't like just fish food or something like that. They have very gourmet tastes. They especially like things like Dungeness crab and stuff like that. So we were all hanging around eating macaroni and cheese and pizza. And, but anyways, it was important and well worth it. This spring spill happened in the springtime when a lot of marine mammals are reproducing and having their babies. This is a picture we're very proud of. This is little baby Ernie. Prior to this time, it was accepted scientific fact that sea otters could not reproduce in captivity. Any attempt prior to this time, the animals had all died. But anyways, the veterinarians at these clinics spent a lot of time trying to study, of course none of us knew hardly a thing about sea otters. There were a few, and they were awfully good, but the learning curve had to be pretty steep. But just logically, they tried to anticipate what would be important for mothers, and one of the things was privacy. And anyways, the good news is we did have many successful births in captivity. We also had many orphans. Many mothers were either died or they were separated from their baby during the capture operation. And so a number of orphaned pups were brought in, and a major effort was made, and very successfully so, to maintain these animals. This again is inside an ATCO trailer. Everything's in an ATCO trailer. This is a waterbed. This is a nice big plywood cage, but down in here is a nice warm waterbed where these little babies could romp and play. And this little girl's name is Princess, and she was learning how to float, and she also liked to get her bath every day, and she'd lay on her back and eat. They're very cute animals, and it's very seductive working with these kinds of animals, but it also is very dangerous, because I'm really not convinced we did them any favors by doing many of these things. After the animals were stabilized in the rehab center and to the point where they no longer needed medical care or attention, they were moved out to these holding facilities. We had 2. One was up in Valdez Harbor at a salmon salmon plant kind of thing, where they they grow salmon. This one was constructed specifically for this purpose. This was in Little Jackaloff Bay down across from Homer, across Ketchmak Bay from Homer. And these were constructed, this is a floating net pen where animals could be housed in groups of about 7. This is a little veterinary shed here. This is a dock where you had a little net where all the floating live Dungeness were for dinner. It was a very elegant facility. So we're going to leave these live animals now and return to them later. In addition to the otters, another major effort was made for birds. I won't discuss them in any detail, but it's important for you to note that major effort was done, and as you can imagine, it was a very difficult and trying process. Very much the same kind of procedures were followed, washing, maintaining them, getting them healthy enough to go outside in outdoor pools. They too had flow-through seawater and had areas where they could get out. They were, as these animals improved enough to be released, they were released intermittently. The sea otters were held until the end of the summer. This is where I spent a good share of my time. This is our very elegant morgue facilities that we had. It's out in the back parking lot of the courthouse, and you can see here that we had 4 lovely garbage cans turned upside down and a piece of plywood. Somehow the pathologists usually kind of get the raw end of the deal here. But anyways, what happened was that all of these people that were out in the Sound picking up the carcasses would send them back in for examination. Most of the carcasses that were found dead in the field were in tough shape. I never truly appreciated the presence of scavengers until I spent the time in Alaska. Many of them were ripped apart, many of them were rotten. They would come in here in either barrels like this or in big plastic bags, be a combination of just about anything you could imagine that they could pick off the beach. They were delivered, this truck would come in, be quite full, and we did with the animals as much as we could. There were 2 categories of people at the morgue facility working on the animals, from the Fish and Wildlife Service and from the Department of Fish and Game. Fish and Wildlife Service people have authority over the sea otters and the birds, and the Department of Fish and Game people were doing the work for their own office as well as for NOAA. So they did basically terrestrial mammals, other marine mammals than sea otters, and other extraneous animals. Like I said, these animals were frequently in pretty tough shape. You couldn't do a lot, but you could at least identify the species, get a general idea of the age, the reproductive status, whatever was possible. These animals were then all tagged and numbered and kept as evidence for the criminal trials that, as it turned out, never did occur. But of the 1,000 sea otters that were examined, all of them were identified and tagged and put in a freezer trailer. Tom will probably talk with you tomorrow. Later on the following summer, some people, Tom I believe included, and residents and all from the AFIP went back up to Alaska with more time and energy, took these 1,000 animals out of the freezer trailers and did gross necropsies on them. So a lot of information did come from them. We didn't do that at this time. This is Kathy Frost of the Alaska Department of Fish and Game over in her lovely facility. And this is what it looked like for Kathy and myself. We'd get just this mound of bags, and there might be a deer in one and a porpoise in another and, you know, a wolverine or whatever. By September of that year, the morgue facility for the Fish and Wildlife Service had examined over 1,000 dead sea otters and over 36,000 dead birds, 150 of which were dead eagles such as this. It was a devastating, devastating spill. Like I said, these were animals that were found dead in the wild and brought in, and they were usually in pretty tough shape. If animals were found dead in the wild and were fresh enough to warrant a full necropsy, or for the animals, the sea otters were brought into the rehab facility and didn't make it, then we did a full necropsy and did it to the best of our ability. We had 2 necropsy laboratories, and they worked out really very well. This one was in Valdez. It had been a salmon processing plant that was taken over, and it came with the metal tables and the hoses and the whole business. it worked out very well. Down in Seward, of course, there was nothing. So again, another ATCO trailer and talking with some good Alaskan carpenters. And I don't know how many of you know Keith Harris, but probably not. Those of you who do probably don't really recognize him here. This is Keith Harris from the Armed Forces Institute of Pathology working on some birds. The facility actually worked out very well. And I wanted to share this type of thing with you today because as a group of pathologists, it was fun. It was frustrating, but it was amazing to see what you could do kind of in the middle of nowhere, kind of with nothing, but with people who were willing to work with you and plumb things in and build things. And it worked out really very well. We had kind of a rule that if an animal needed to be necropsied, if it weighed under 250 pounds, it was brought into us. If it weighed more than 250 pounds, we had to go to it out in the field. And I'll have to admit, we even had a little bit of fun while we did it. We had a NOAA helicopter at our disposal for this. And we had necropsy kits ready to go in coolers. And then we were taken out into the field where we could get to the animal. This was a fully grown, full-term pregnant sea lion. In the field, you can't do a lot. The weather is frequently a serious problem, too. But we were able to do a gross necropsy and then at least take out her major organs, one lobe of lung, one kidney kind of thing, put it in the coolers, and take them into the lab and deal with them from there. This is a little bit of my philosophizing, but one of the problems I had with the way Exxon handled the event was that in mid-September, they determined that the event was over, essentially, and that they had cleaned up everything they were going to clean up, and that it was done, and we were all going to go home and forget about it. But by this time, I was on assignment to the U.S. Fish and Wildlife Service, and in fact, we were having a large number of animals still die. Unfortunately, one day I went into work, and Exxon had shut down the laboratory facilities that we'd been working out of. So we were kind of on the street. But anyways, we had a wonderful opportunity. They made the Scientific Crime Detection Laboratory in Anchorage available to us for the rest of the fall. So I was able to complete my necropsies in there, and it was the most elegant facility I've ever worked in. To give you an idea of the types of many types of animals, in addition to sea otters that we necropsied, we did many harbor seals. Of course, lots of sea otters, sea lions, porpoise, and whales. When I went there, tasked with working on mammals affected by the spill, I really was thinking marine mammals. But when I got there, I learned very quickly that it wasn't just marine mammals that live in the ocean that are impacted by a spill like this. A great number of terrestrial mammals are affected as well. And these are terrestrial mammals that either take their food from the sea or who come down to the sea and eat scavenge on the beach. So these included animals such as river otters. We saw a great number of these. Also mink. An awful lot of Sitka deer who come down to eat algae in the intertidal area. Coyote, who are scavengers. This little guy didn't have any oil at all on the outside of him, but he had a whole belly full of oiled bird feathers and bird bones. The brown bear was also considered to be at risk. I think the final decision or determination was that they did not suffer during this spill, but since they are both scavengers as well as using the marine waters for their primary protein source, they were considered to be at risk, and they too were monitored. We also saw a great deal of marine birds, such as puffins, and other species that I really wasn't familiar with at all. All kinds of things came in dead. How much of this was opportunistic, we don't know. Bring my slides off just a little bit there, but I also wanted to bring home the point that it wasn't just sea otters who were having their babies during this time. We also saw, unfortunately, a number of other types of fetuses or neonates. And that included sea lions, porpoise, many, many sea otters, and harbor seals. As you can see, this is a very brand new harbor seal, just a fresh umbilicus there. And in a little while you'll see, for those of you who aren't familiar with harbor seal pups, this is literally a tar baby. They're really supposed to be almost a white color, at least a light brown. But when we got these dead ones, they were just literally tarred. As Dr. Nielsen mentioned just a moment ago, excuse me, about the gross lesions, and I think Tom will show you more about this today, but it was so exciting I just have to show one too. And that is one of the prominent lesions of these animals was the emphysema in their lungs, and it was extremely prominent. You can see these large areas of bullous emphysema here. And one of the things that was so interesting about this was before I started doing the necropsies, many of the clinicians would describe these animals coming in, and they had all this crepitation around their chest area, and they really couldn't figure out why. But anyways, when we did the necropsies, it appears that the bullous emphysema, came right there at the hilus of the bronchus, excuse me, right up big pockets of air all the way up along the trachea, through the thoracic inlet, and then down into the subcutaneous tissue. It was really quite remarkable. I think Tom is going to be covering, excuse me, the majority of the lesions tomorrow, but I want to show you just a few that I find particularly dramatic and very characteristic of what I saw. The most common thing that I saw was hemorrhagic gastroenteritis, leaving very, very pale intestinal tract like that. And then I wanted to focus on the fact that though a lot of good was done for these animals, and I think everyone did everything with the right intention, with very good intention, still we managed to kill a number of animals by accident. One of the things that was a problem at the beginning was in animals during the capture process. Some people, I think, were a little too gung-ho, not very knowledgeable in the biology and the activities of these animals. And there were a number of incidents where mothers were giving birth to their babies or about to give birth to the babies out in the natural environment. And of course, she's not really swimming around real fast when she's doing that. And so these people in the boats would see this otter not swimming very fast, and then they'd chase it a little bit, and she couldn't get away very fast. And so they'd chase it a lot, and they would sometimes, as in this case, basically chase it to death. This was a female otter that came in, obviously pregnant. What's very, very dramatic about this animal was, to me at the time, was this beautiful layer of fat, which animals that died after exposure to oil did not have. They became very, very emaciated very quickly. She had a very nice fat layer. She had the most elegant silken coat I've ever seen. She was at full-term pregnancy. It was torsed here right at the base and was completely necrotic. It appeared that she had been chased and either torsed during the chase itself or when they brought her on board. Another case of, again, doing everything right for all the right reasons and things still go wrong, and God knows that's a law of veterinary medicine. This particular animal was in the rehabilitation center and was most likely dying anyways, but they needed to take a blood sample from his femoral vein, and unfortunately the animal still had enough strength to struggle, and the femoral artery was lacerated in the process, and the animal bled out. So well, I think it's important to bring this type of thing to your attention as veterinarians and as people who may be in positions of being involved in making decisions about future capture and rehabilitation activities. Just very briefly, we've all talked about the routes of exposure to the animals, but this is a slide that I wanted to make sure that you saw. This really brought it home to me how really tenacious and thick and devastating this oil is. And the slide you've already seen, this is not your typical car oil that is spilled when Prudhoe Bay crude goes into the water. In fact, for those of you who've been watching TV in the hotel this week, they're making a lot of big hoopla up in Valdez Harbor again with that minor oil spill that they've had up there. I think they may have even dug out some of the old footage because it looked awful familiar. I'd like to move off of land now and out of the necropsy lab out into Prince William Sound. Well, while we were all doing all of our stuff back there in the laboratory, there was still a lot happening out in the field. And this has to do primarily with harbor seals. The National Marine Fisheries Service, in conjunction with the Alaska Department of Fish and Game, made the decision not to mount a capture and rehabilitation exercise like the Fish and Wildlife Service did for the sea otter. They did this for a number of reasons, and I think probably a number of very good reasons. As we've talked today, the harbor seal is not as much at risk to oil as the sea otter is. There's a good, healthy number of animals up there. They felt that they could do as much harm as good by going out there, running around, separating moms and babies. They couldn't bring mom in because she's just too big. There's just no way you're going to be hauling 250-pound mamas and their babies in. So for a number of reasons, the decision was made to not bring them in. Since this decision was made, however, it was also a very important priority on the part of all of the federal and state agencies to get to the pupping areas and get them cleaned up of the oil as quickly as possible. As the oil spread out through the sound, it hit many of these known rookeries that the harbor seals used. So it's well studied historically that the harbor seals start to pup at about day such and such. And we had about 3 weeks to spare. So they did, in fact, working with all the appropriate people and committees and all that sort of stuff, set the priority for beach cleanup as the beaches or the rookeries where the animals would be using for pupping activities would be cleaned first. And in fact, this did happen. Excuse me, I meant to comment on this. This is a typical area where harbor seals will pup. You can see a number of harbor seals here. Normally, moms are in the water almost all the time, but when they do have their babies, they crawl up onto these little rocky ledges, have the baby, leave him there for a few days, they go out, they eat, come in, take care of the baby, and everything is fine. As you can see, this oil sheen coming off of this rookery, this one has been oiled. To put this in context, these slides, by the way, were provided to me by Lloyd Lowry and Kathy Frost of the Department of Fish and Game. And they took these out on Night Island. And Night Island is really craggly little inlets, and the way it's geographically located, the oil would come and it would hit some and it would just miss others. So sometimes animals could be 50 yards away as the crow flies, and some would be in completely contaminated areas or lagoons, and the other ones would be in clean water. This is what a mama and baby harbor seal are supposed to look like, and that's what they did look like in the one arm on Night Island. Next to that, this mother had her pup in an area where it was oiled, and you can see the oil starting to accumulate here on this pup. And I think this slide is one of the most meaningful that I brought home from this experience. And this is a harbor seal pup, obviously completely oiled. But what's so fascinating about this slide is that this beach has been cleaned already by the cleanup crews and had been named environmentally stable by all of the appropriate officials, and everybody thought everything was hunky-dory. But the reality is that this plant material, and I think it's called fucus, is very spongy-like. So you can clean the outside of it, and it really does look pretty clean. But when a little baby seal lies on it, all of the oil squeezes out of it, and he becomes oiled anyway. We kind of lost the tops of these guys' heads, but in addition to the oil impacting the animals, there were a lot of other things going on out there in the sound that didn't have anything to do with the oil itself and the toxicity of the oil, but had to do with the cleanup effort. Prince William Sound usually is essentially uninhabited. There's a few little hunting cabins out there, and some people go out and kayak, and boats come and go. But it's essentially a very quiet, very pristine kind of place. When all this happened, on any given day during that summertime, there were approximately 10,000 people out in the Sound cleaning up the beaches. And they brought with them to do their job all of their equipment to clean the beaches, the high-power hot water washers, literally the armadas of boats and landing gear and planes and helicopters that they need to transport both the equipment and the men. Because, as we've said, they can't just go back to Valdez Hotel at night, they had to bring their own living quarters with them. And so barges were brought out and echo trailers stacked up on top, and the men and women of the cleanup crews lived here, and then boats brought out major supplies, and airplanes would ferry people back and forth and around. As you can imagine, this was an enormous amount of activity going on out there. And there was a lot of concern within the wildlife community that all this hoopla may very well impact the animals as well. And that's a very difficult thing to prove. But there was one species that we were able to document very well. The Fish and Wildlife Service has historically followed eagle nests for years, and they know where the nests are, they know what years they're productive or not productive, and so they followed these, and they found that in the areas of high beach activity there was a 71% failure of the eagle nest that year. I'd like to go back now to the animals that have survived the rehabilitation and that are now floating around eating Dungeness crab and gooey duck and having just a marvelous time. It was a big deal to decide what to do with these animals. They needed to be released back into the environment, but they didn't want to release them onto the western parts of Prince William Sound where the oil may still be. And so they had to, the Fish and Wildlife Service was responsible for finding new habitat for them on the eastern side of the Sound. And so of course you have to hit the criteria of, you know, what they need to eat and all those things. They also had to take into consideration the native communities that were in the area. It seems like everybody down here in the lower 48 just loves these cute little brown things. But the reality in Alaska is that they are competitors for food with the native subsistence hunters, and they are very much hated. So another priority was to find habitat where these animals could be released that did not conflict with the native Alaskans. So anyways, all this was done. During this time, close to 400 animals were brought in. I think it was like 390 or so. Depends on how you define it to an extent. And about a little over half of those survived to the end of the summer. And were almost all of those were then termed releasable. There were a few animals that needed to go to aquaria, and those were animals that were either too old, a few had some permanent medical problems that they could not be released and expect to survive. And then, of course, all of the orphans had to go to aquaria since they couldn't survive without the mother. But other than that, the animals were to be released. During this time, an immense amount of energy had gone into rehabilitating these animals. It was an fantastic opportunity to be able to study these animals, but also a lot of work on the part of hundreds of people. The dollar figure, I'm not exactly sure. Back at the time of the end of this summer, it was estimated to be approximately $8 million that was spent by Exxon Corporation on the sea otter. I have since heard a representative of theirs now say that it's closer to $18 million. So I really don't know, but it was a lot of money. So as part of all this, we wanted to know, we from the Fish and Wildlife Service wanted to know whether this was a good thing that we had done. Was it a worthwhile thing for the animal? And will the animals benefit from it? So prior to release, we wanted to be able to tag the animals so that we could follow them and evaluate their survival to the best of our ability. That was another political hoopla with the environmentalists from hell who didn't want us to do anything to their little otters because they'd already been traumatized enough. But we did get some compromises, and so minimally all sea otters that were released were flipper tagged, and then a subset of animals, and I believe it was 45 total ended up, had radio transmitters implanted in their abdomens. Unlike other species of marine mammals where you frequently hear about radio tag collars and things on their dorsal fins and all, it doesn't work on the sea otter, they found out over the years. It needs to be implanted. It's a free-floating object that goes inside a midline incision, and I'll show you in a moment. But anyways, 45 animals were selected, some of them here at the Valdez Center and some down in Seward, or at the Jackalope facility. I'd like to show you just a few pictures, which I found, it was a fascinating event, again, to try to figure out how to do something in some fairly unusual circumstances. These animals were outside in the middle of Valdez Harbor, so we couldn't really very well pack up a bunch of animals and bring them over to the local clinic. So instead, some of these great Alaskan guys built us a lovely surgical suite on this floating dock out next to the floating net pens where the animals were kept. We brought all of the equipment out, and a team of people. For those of you who know Tom Williams from the Monterey Bay Aquarium, is the person who has basically worked out these techniques while working on animals in California, and he spearheaded this effort. In addition to doing the surgeries on these animals, other information was collected. Teeth were collected for aging. Measurements were taken. We tried to gather as much information to characterize the individual animal as possible. In addition to the radio transmitter, these little barcode dealy guys, I've forgotten the real name for them, were implanted just subcutaneously in the groin area, and they will stay with the animal or the carcass forever, so that they can be read digitally to give a specific identification to the animal. This is a picture of the transmitter that actually goes inside the abdomen. It's about the size of a pack of cigarettes, and it's a non-reactive silicone-type material. One of the things that I found fascinating was we go through all this stuff with this animal that absolutely cannot have anything done to disrupt the insulation provided by its hair coat, or it will die. Well, then how do you do surgery? You know, where I came from, you always do this real nice clip job and you scrub and everything. But anyways, again, Tom had figured this out. They scrub it. They scrub all of the hair very nicely and cleanly, and then they put KY jelly on it and that's a comb. Take a comb and part it right down the midline, paste it down, and then put a plastic drape on it. Worked beautifully. And it's a very simple procedure. You just open it up, pop it in, and sew it shut. For this surgery, also another thing that was important was to be able to, as my boss told me, don't let any of these animals die. It was a very important thing to preserve the animals, but also from the public relations standpoint, it was brutal. What was done with these animals for anesthesia was we used an injectable narcotic and then used an antagonist to reverse it, and literally within 1 to 2 minutes, the animals were back in the water. We had a complete success, and it was a fascinating experience. So then we packed everything back up in the boat and went on and went down to do the rest of the surgeries down in Little Jackaloff Bay. And anyways, it was finally now fall, and it was time to release the animals according to the release strategy. So the animals were loaded back into their dog carriers, loaded onto big helicopters provided to us by the Coast Guard, and they were flown over into the pristine areas of the eastern part of Prince William Sound that had never been oiled. And then it was finally time. It was the day for the release. You can see this little guy in here, and he's just happy as he can be, and it was beautiful. This is, for those of you who are interested in, this is the little electronic gizmo, the wand, and the digital readout machine that reads those barcodes. This is used, I understand, in many species of animals now, and this is where you'd see the digital readout. So it's finally time to let them go, and it was great. I'll have to admit, for as depressing as the summer had been, it was finally truly wonderful to, at least for a few animals, to open the door and see them head out and catch their buddies, and they knew exactly what to do. And while they were all frolicking around out there having a marvelous time back in the natural environment, the Fish and Wildlife Service biologists were heading to work. You can see this boat here is outfitted with the radio antenna for tracking, but in addition to just tracking these animals, a number of studies was conducted to follow the success of these animals, to monitor for reproductive success, for any kinds of other disease processes that may be going on, a whole host of things. So they went to work. So in conclusion, I'd like to say that it was a tremendous experience. I think we did a lot of very good things, but I think that this type of work, and especially the science that comes out of this, is very crucial, because I think we need to use this type of science to hopefully be able to be about the only ammunition we may ever be able to have to improve our laws or our regulations to improve the safety. Another spill or another accident will happen, that's guaranteed. Not sure you can all see exactly what this is, but we've been seeing it on TV the last couple of days. These are the tanks of the Valdez terminal. This is the dock here that we've been seeing on TV. And this is the resident population of sea otters that live right in Valdez Harbor. I'd like to see this kind of science continue, and I'd like to see it really put to use, because I've got one more story I'd like to tell you. And this is about this big old guy that was brought to me in the fall. But the story behind him is one that absolutely just about broke my heart at the end of the year. He was a big, strong male, and he was found down in Windy Bay early in June. And he was fine. He wasn't oiled. There was no oil even in Windy Bay yet. But they knew the oil was coming towards Windy Bay, and so they thought to preempt any exposure, they would capture this animal, which they did, and they brought him into the Seward Otter facility, where he swam around and ate all that stuff and made new friends and had a marvelous summer. And he maintained a very good health status. At the end of the summer, he was loaded into that helicopter along with the rest and flown over to the eastern side of Prince William Sound. But he is a big dominant male, and as soon as he was released, he hit the deck and he swam right straight across Prince William Sound back over to Windy Bay. Windy Bay, in the meantime, had also been termed environmentally stable. It was completely clean, it was stable, and everybody had gone home. When he got over there, they found him 48 hours after his release, and they brought him into me. He was as oiled and as tarred as any animal I've ever saw early in the spill. So what that really brought home to me was that this spill happened in the spring, when the weather was absolutely as good as it gets in Alaska. We had all summer to work on it. Unfortunately for Exxon, but fortunately for the country, it happened to probably one of our wealthiest corporations who were willing to put the money and the effort into doing, I think, everything humanly possible to clean up the spill. But the bottom line is that when you do something this devastating in the marine environment, you just, no matter how hard you work or how much money you're willing to spend, you can't change it and you can't fix it. Prince William Sound isn't going to go away, but it won't be quite the same. And I hope that we can continue to use our science to prevent events like this. Thank you very much, and let's take another break and come back and talk about birds in a little bit. Again, in case they listen to this tape. I'd like to acknowledge the tremendous help of Mark Pokris and Milt Friend in helping me put this talk together. Mark is with the Wildlife Clinic at Tufts Veterinary Medicine School in Massachusetts, and Milt, who I'm sure many of you know, is the director of the National Wildlife Health Center. In fact, my understanding is that he essentially created it, and I think maybe he invented birds too. I'm not too sure about that. But anyways, much of what the slides and the graphics that I'll be showing you today have been provided to me by Milt and Mark. as a way of introducing the talk today, I'd like to show excuse me, some of you some examples of some of the species that I'll be talking about. And I'd like to include in this talk both truly marine birds, but the diseases aren't specific to either those or the coastal birds or our waterfowl. So I'd like to at least show you some pictures of some representatives of all of these different species. This is an albatross, who is a Pacific inhabitant. Greater shearwater, who's out in my type of ocean. We'll also be talking about our coastal birds. Many animals who feed from the sea also live in our very coastal environments. Some of them are endangered, like the brown pelican, and these are very important animals for consideration. Also, we'll be talking a lot about our real common inhabitants of the shorelines, such as the cormorants and the gulls. These guys can be very important in not only diseases, but in disease transmission sometimes. Also, we'll be talking a lot about animals like geese and swans and ducks and other waterfowl. We have an awful lot of these up in my area. They're spectacularly beautiful. But it's also important to keep in mind that there are marine birds, so to speak, in many parts of the world that we really don't see, that are very unusual, and we need to incorporate those in our thinking of marine diseases. I'm going to try something a little different in this part of the talk. I'm going to use two projectors here. So I'm going to try to remember which is my right hand and left hand and stuff like that. The way I've divided the talk up is I've tried to categorize the diseases of marine birds into the pockets of how they are caused, or what they're caused by, the etiologic agent. Many of these topics have been covered to some extent or another already. Certainly Dr. Nielsen talked about some in the last hour. So when we get to those areas, I'll briefly comment and then move on. So I'd like to start out first in our discussion with bacterial diseases and start with avian cholera. Avian cholera is a highly infectious disease caused by the bacterium Pasteurella multocida. Transmission may occur by either bird-to-bird contact, ingestion of contaminated water, and also possibly through aerosol form. What in the world happened? Oh. I'll just take the, excuse me, that got in the wrong place. It wasn't at the beginning. The species affected by avian cholera, many of the scavengers are particularly involved. But it's important to note that prior to 1944, cholera was literally unreported in free-living birds in the United States. Since that time, losses have now been reported from coast to coast, and the occurrence has risen dramatically since 1970. Avian cholera now ranks with botulism and lead poisoning as a major cause of waterfowl mortality. Within wild birds, cholera is very common in waterfowl, and as already mentioned, particularly in scavengers such as gulls. In the field, occasionally very few sick birds will be seen because death can occur very acutely and you may only find carcasses. If birds are alive, they may show dramatic evidence of CNS signs, including convulsions, circular swimming, unusual head postures, as seen here in this bird, who's holding his head back in a very stiff position. There also may be some signs of erratic flying behavior, some mucus discharge from the mouth or around the eyes, bill, or vent. The droppings may be unusual. A number of things and a lot of variable signs, but these field signs are not necessarily unique to avian cholera. Some of them are similar to duck plague or some of the other diseases we'll be talking about later. I think it's important to always suspect avian cholera, though, when you see large numbers of dead birds in good flesh, and these birds are found within a short period of time, which means that they're dying suddenly. In fact, death can be so acute in this disease that literally birds can fall out of the sky dead. Grossly, usually the carcasses have substantial amounts of cutaneous and visceral fat. The most prominent lesions affect the liver and the heart, Hemorrhages occur on the surface of the heart muscle, as seen here. There also can be focal necrosis of the liver. And in the GI tract, there can be these thickened yellowish areas that are filled with fluid down in the lower tract somewhere. And these fluid areas are literally chock full of the organism Pasteurella multocida. The occurrence of gross lesions is dependent on how long the bird lives after infection. Diagnosis of avian cholera is dependent on either virus isolation or viral culture. Avian botulism is a paralytic, often fatal disease resulting from ingestion of the toxin produced by the bacterium Clostridium botulinum. 7 distinct types of toxin have been identified and are designated A through G. Waterfall die-offs and botulism are usually caused by either the type C toxin and sometimes by type E. Factors leading to an outbreak of botulism are not fully understood, but it appears quite clear that when an outbreak does occur, it's usually perpetuated by a bird-maggot cycle. In the wild, many species of waterfowl and shorebirds are affected by botulism. And avian botulism is one of the 3, and perhaps now maybe one of the most important diseases of all time in migratory birds. Important environmental conditions that contribute to the initiation of avian botulism outbreaks include water depth, water level fluctuations, and water quality. The presence of vertebrate and invertebrate carcasses, rotting vegetation, and high ambient temperatures are very relevant to the onset of this disease process. Because these optimal conditions frequently occur along the shoreline, this is also frequently where the carcasses are usually found. If birds are seen alive, there may be paralysis of the voluntary muscles, resulting in an inability to sustain flight. There also may be paralysis of the nictitating membrane or paralysis of the neck muscles, resulting in an inability to hold up the head. Death in this disease is usually due to drowning or to respiratory arrest from the toxin. There are no characteristic gross lesions in waterfowl associated with either type C or type E botulism. Presumptive diagnosis of botulism can be made from a combination of signs in sick birds along with the absence of lesions in dead birds, but for a definitive diagnosis, the mouse protection test is recommended. Numerous outbreaks of avian botulism have been associated with sewage and other wastewater discharge into marsh environments. This relation is not clearly understood, but it's occurred often enough that wetland managers should really discourage the discharge of any effluents when substantial waterfowl or shorebirds are using these areas. Avian tuberculosis we've already heard about, so I'll speak to it very, very briefly. It's caused by Mycobacterium avium, and I think we'll get that cleared up there in a second. It affects many species of birds, man, and many other mammalian species, and the highest prevalence is in scavengers such as gulls. Advanced disease and clinical signs are seen most often in the adult birds due to the chronic nature of this disease. Field signs of avian tuberculosis, there are no specific signs, but birds may appear emaciated, weak, lethargic, and there may be a variety of lesions or there may be none. These lesions that may occur include very generalized types of things like diarrhea, lameness, etc. Gross Grossly, the carcasses are usually very emaciated, and they have crumbly yellow-white nodules in the infected organs. The liver is the most commonly affected organ, as shown here. Also, the spleen may be involved, sometimes the lung or the intestine. These lesions are raised, firm nodules, and it's important to remember that these nodules look very similar to those found in aspergillosis. There are some very important environmental considerations of avian tuberculosis. Fecal contamination is the major means of dissemination, and it's important to remember that this organism can survive for a year or more in organic substrate outside the body. Chlamydiosis is a disease caused by Chlamydia Cetacea, a rickettsial-like intracytoplasmic organism. Though chlamydiosis is usually associated with disease in cetacine birds, domestic fowl, or feral pigeons, it's also been reported in 140 species of wild birds. Among the reports in the wild birds, many of these are in shorebirds and in waterfowl. Whoops. The gross lesions of chlamydiosis, frequently there is evidence of splenic enlargement, which I do not have a picture of, but there commonly also is a serous exudate on the heart, as shown here, and this serous exudate may also include the air sacs. The liver frequently is also involved as well, and the liver may be enlarged with areas of necrosis and a thickened capsule. There are some human health considerations that need to be remembered about chlamydiosis, and that is that you can get sick. There have been reports of wildlife biologists becoming quite ill with chlamydiosis following handling of snow geese, great egrets, ducks, or winged doves. Avian salmonellosis is really a group of diseases. All of these diseases are caused by organisms in the genus Salmonella, and there are somewhere between 1,800 to 2,000 serotypes of this disease, of this organism. Para-Typhoid infection is the disease caused by Salmonella typhimurium, and it frequently causes illness and death in wild birds. The transmission can occur via many routes, and die-offs frequently occur in mute swans, ducks, and in coots, American coots and gulls. Grossly, the occurrence and type of lesions are extremely variable. Acute cases may have no lesions at all, or the para-typhoid lesions in the body and the cavity may be small and white and tan and granular. There have been reports of arthritis in the hips and knees of domestic ducks. There are also important environmental considerations associated with para-typhoid infection. Point sources of contamination may frequently be the source of these outbreaks. Point sources may include either refuge dumps, sewage wastewater point sources, or point sources or runoff from either livestock or poultry operations. These are very important sources of this infection, and they also should be kept in mind for wetlands planners. I'd like to move on very briefly to viral diseases now. This is a list of common viruses in birds in general, and I'll be speaking only to avian pox this afternoon. And of course, that's already been discussed by Dr. Nielsen, so I'll discuss it even more briefly. Avian pox is a mild to severe, slow-developing disease in birds caused by a large virus belonging to the aviopox viruses. Transmissions can occur via mosquitoes or by contact between contaminated surfaces and abraded skin. Though avian pox infects many species, the cases in wild birds are quite limited. However, within the wild birds that are affected by avian pox, the albatross and the murre are two species that have had large numbers of animals die. For field signs of avian pox, birds with wart-like nodules on one or more of the featherless areas of the body, such as the feet, the legs, the base of the beak, or the eye margin, should be considered suspect cases of avian pox. Even though the course of this disease is long, some of the birds have been known to recover from it fairly well as long as the nutrition is maintained and secondary infections are controlled. Grossly, there are two basic forms of this disease. The external form has warty nodules forming on the exposed surfaces of the mouth and the legs, as already described. Usually these can be quite small, but if they become large enough, they can actually destroy the animal's ability to see or to eat. The less common form of this disease is the diphtheritic form that affects the mucous membranes of the upper digestive tract as well as the respiratory tract. This form of disease has been reported only in a few species of birds, including the mute swan, the layman albatross, and the common murre. Diagnosis of avian pox. A presumptive diagnosis can be made from the gross lesions alone, but a definitive diagnosis needs to have identification of the characteristic inclusion bodies on histopath and or virus isolation and serological identification. Aspergillosis is a fungal infection of the respiratory tracts of birds and mammals. As, again, this disease has been discussed briefly already. Aspergillus is commonly involved in many avian species and frequently occurs in waterfowl and loons. The young are often more susceptible than adults, and frequently we see this disease as secondary to another stressor. And the other stressor may be either another disease process or an environmental stressor such as captivity. As I've already mentioned, this disease occurs frequently in our waterfowl as well as many of our other birds. In the field, the typical signs of aspergillosis, the animal is emaciated and frequently manifests either severe or progressive difficulty in breathing. And this is characterized by the mouth gaping type of posture that this animal is showing. Birds often appear unthrifty, and they may have a wing droop where their wings just kind of hang like that. These kinds of field signs, though, are not necessarily specific to aspergillosis, and you may see these in several other diseases. In acute aspergillosis, the lungs are dark red and firm with 1 to 2 millimeter yellow nodules, and the air sacs can be thickened. In chronic aspergillosis, the lesions in the lungs and air sacs become more dramatic, like shown here, and the yellow plaques become cheesy-like in appearance, and there may even be, a soft, fuzzy, matty growth on these lesions. I'd like to move on now to parasitic diseases, and of these I'll speak only to a couple, to Sarcocystis, to gizzard worms, and to nasal leeches. Sarcocystis is a non-fatal, usually asymptomatic parasitic infection caused by a protozoan. The infection occurs when waterfowl ingest food or water that contains the egg of the parasite. The species affected frequently include the ducks, the geese, and our swans. The gross lesions of Sarcocystis are marvelous to find. They're very easy. The visible forms of the disease appear just as you remove the skin. In the breast muscle, there are these creamy colored little cylindrical cyst forms in the breast muscle. Sometimes there'll be some calcification around it and that will give a gritty kind of feeling when you cut through it with a knife. The life cycle of Sarcocystis goes through a carnivore. Gizzard worms. Several species of nematodes and migratory birds are referred to as gizzard worms. Two most common are listed here. These long, sometimes coiled, thread-like worms are located just beneath the surface of the grinding pads of the gizzard and are most frequently found in waterfowl. Like I just said, this occurs very commonly, especially in our geese and the coots and some of the ducks. The life cycle of this organism is direct. We don't need an intermediate host. The gross lesions are important because there are no field signs. In the field, there are no specific signs related to this parasite, but the heavy burdens may cause the animals to basically be unthrifty. There may be poor growth in the young and some emaciation in birds of all ages. If infections get to be really severe, it may interfere with food digestion, and as a result, there may be extensive damage to the gizzard. But when the bird is opened, grossly, we can see a number of changes. In the gizzard itself, there's inflammation, hemorrhage, and ulceration of the lining of the gizzard. There may be worms actually present, as you can see up in this area, these tiny little thread-like worms. Also, there may be necrotic areas in the gizzard muscle, as shown here, due to migration of the parasites. Is anybody's eyes getting tired yet? Blood-sucking leeches of the genus Theromyzon are the only leeches in North America known to feed directly in the trachea and the nictitating membrane of migratory birds. These leeches normally affect water aquatic species, and especially those birds who live in the north. This parasite is particularly well adapted to the cold waters and tends to affect our northern birds with peak parasitism usually in the spring and the summer months. As I said, the relative frequency is most commonly found in the ducks and in the swans. In the field, these leeches are really disgusting. They may be grossly visible attached to either the nares or the mucous membranes. If you see this in live animals, the animals are likely to be fairly uncomfortable and be shaking or scratching their head or sneezing. And if the leeches have gone inside into the breathing passages, the animal may be showing labored breathing or gaping. These leeches are really big and disgusting, as I said. And depending on where they attach, they can cause trauma. If they attach in the eye area, there obviously would be trauma there or inside the nasal cavity. Sometimes these can be so bad that they can actually go way up into the nasal passages and actually cause a blockage. For environmentally associated diseases, I think that my marvelous colleagues have really preempted me on this, and I think that I'll just barely skim through it. On Sunday, Dr. Blodgett spoke very eloquently on the chlorinated hormone disruptors and the many problems that they may or may not cause, although it's looking much more like they may. He cited some references, and I have one of those with me, and I have a couple of other suggestions too for those of you who are interested in it. Dr. Nielsen has recently spoken to lead shot and sinkers and oil toxicosis, so I'll go through that very briefly. In terms of debris, that is persistent marine debris, which we mentioned in the last hour, and the land-based contamination, this is not a specific problem, but it's important, I think, to keep it in light of this environmentally associated disease, many of our pathogen-associated diseases. One of the problems we see here particularly are problems with salmonellosis from land-based refuge dumps. There's becoming more and more a problem in our Rhode Island area where garbage refuge dumps are near the ocean, all kinds of trash there. And either the birds come in and can get salmonella themselves and become ill, or they can act as a vector. Last fall, I participated in a course at Guelph at the veterinary school on ecosystems health. And one of the case studies that was used there for the students was an actual case of which it hasn't finished, so I don't know the end of the story. But there was a situation where there was the town refuge dump for Guelph, and across the road was a dairy farm. And over time, the dairy cattle just kind of went downhill. Production went downhill, and they started to get sick, and then they died. And eventually the man had them all killed off. But he felt that the cause was from the dump across the street. So there's been a lawsuit, and the students were working on trying to do just an intellectual study of this, where did the birds bring salmonella across and, you know, with it on their feet and get it in the water source? Did other organisms come through drainage through the groundwater and up? There's a number of diseases and problems that haven't been looked into in light of that type of a problem. So I mention that just for something to think about and talk about over a beer sometime. Lead poisoning is an intoxication resulting from absorption of hazardous levels of lead into the body tissues. In our waterfowl, by far the most common source of this lead is lead shot, but there are also other sources of lead, including fishing sinkers, mine wastes, paint pigments, bullets, and any other type of lead that an animal can get in their mouth. Lead poisoning has affected every major species of waterfowl in North America and has also been reported in a wide variety of other birds. Within the United States, the annual losses in the last few years have been estimated to be between 1.5 and 2.5 million waterfowl per year dying from lead poisoning. With the use of non-toxic lead shot, these numbers should be coming down considerably. Lead poisoning is most common in our ducks and geese, and in other species of birds, the incidence of lead poisoning goes down as their specialization of their dietary sources on food increases. So in our shorebirds, we certainly do see it, and it's also seen in the raptors. Raptors may ingest lead shot in ingesting carcasses that they find dead on the beach that have been killed by other hunters. To just very briefly go through the field signs of lead poisoning, usually the animals may be reluctant to fly. If they do attempt to fly, usually they cannot fly for very far. They don't fly very well. They may have unusual head postures that may be bent or crooked. When they land, they don't do it very gracefully. And eventually, as the disease progresses, they may not be able to try to escape at all. They may have some discharge from their fluid discharge from their mouth as well. Grossly, what we see are signs of emaciation where there is tremendous wastage of the breast muscle. There may be significant bright green diarrhea around the vent. As Dr. Nielsen mentioned, swelling in the head due to fluid accumulation. Changes in the GI tract leading to impaction. And certainly presence of lead pellets throughout the digestive tract. There may be many, and as Dr. Nielsen said, these may be partially absorbed. They may be in various sizes and shapes. In addition to changes in the gizzard and the proventriculus, there also usually is a distended gallbladder that is dark in color and filled with bile. It's important to note, however, though, that if animals die very acutely from high levels of lead, you may see few or none of these types of lesions in the animals. As far as control of lead poisoning goes, when it is caused by lead shot, really the only thing that you can do to prevent this is to basically outlaw non-toxic or outlaw lead shot. I'd like to comment on the tremendous amount of work that Milt Friend has done in using the science developed by himself and his laboratory and others, and basically tackling all the political brouhaha that you have to get some changes in regulations, and that has led to the use of lead shot, non-toxic shot, and hopefully for the protection of these animals. One other incident
of lead poisoning that has really come to the forefront is in the loon. Mark Pokris at Tufts has done quite a lot of this work, and they find that these animals ingest sinkers from lead sinkers from fishing line. Obviously, lead is lead, and it causes a syndrome essentially identical to that seen in the waterfall from lead shot. Again, I'd like to applaud Mark for his work and his collaborators. They have taken their science and apparently the causal relationship that they've been able to show, and they've even gotten EPA, which is a really difficult thing to do, is they've gotten EPA to move and take action on this. And there's currently in the Federal Register a proposed rulemaking for doing away with lead sinkers as a result of this type of work. And very briefly, the last comment, I'm sure you probably all want to hear on oil for a while. Birds exposed can be damaged due to either hypothermia, they can ingest oil, they can inhale it, or they can absorb it through their skin. Via any of these routes, animals can be severely injured. All animals, all birds essentially are susceptible to damage from oil in the marine environment. It really depends on what bird happens unluckily to be present when a spill occurs and how they use their habitat. There is some difference in the severity of the damage to a particular species based on some animals. Oops. The puffins and the mewers are birds that have very long reproductive cycles. So if they are to be hit hard in an oil spill like they were during the Valdez spill, it may literally take them decades to get their population back up to pre-spill levels. Also, other birds like the brown pelican is an endangered species and is at special risk from oil spilled into the environment. A comment that was made, I believe, by Dr. Zinkel a few days ago is a reminder that not all oil spills happen in the marine environment. And in fact, many of them, probably the majority of them, do not. Many happen inland or in freshwater river systems, around industrial plants, whatever. However, when they do happen in the marine environment, especially if it's a very large spill, it can be very devastating. And as Dr. Nielsen talked at length, the gross lesions that are caused by exposure to oil in the marine environment to these birds are many. So I'd like to close there and thank you very much for your time. And it's been a real pleasure to talk with you this afternoon. And hopefully we can all continue to work so that we can use our science to keep our little guys out there in the marine environment healthy and happy. Thank you very much. Marvelous time. It was a big deal to decide what to do with these animals. They needed to be released back into the environment, but they didn't want to release them onto the western parts of Prince William Sound where the oil may still be. And so they had to, the Fish and Wildlife Service was responsible for finding new habitat for them on the eastern side of the Sound. And so of course you have to hit the criteria of, you know, what they need to eat and all those things. They also had to take into consideration the native communities that were in the area. It seems like everybody down here in the lower 48 just loves these cute little brown things. But the reality in Alaska is that they are competitors for food with the native subsistence hunters, and they are very much hated. So another priority was to find habitat where these animals could be released that did not conflict with the native Alaskans. So anyways, all this was done. During this time, close to 400 animals were brought in. I think it was like 390 or so. Depends on how you define it to an extent. And about a little over half of those survived to the end of the summer. And were, almost all of those were then termed releasable. There were a few animals that needed to go to aquaria, and those were animals that were either too old, a few had some permanent medical problems that they could not be released and expect to survive. And then, of course, all of the orphans had to go to aquaria since they couldn't survive without the mother. But other than that, the animals were to be released. During this time, an immense amount of energy had gone into rehabilitating these animals. It was a fantastic opportunity to be able to study these animals, but also a lot of work on the part of hundreds of people. The dollar figure, I'm not exactly sure. Back at the time of the end of this summer, it was estimated to be approximately $8 million that was spent by Exxon Corporation on the sea otter. I have since heard a representative of theirs now say that it's closer to $18 million. So I really don't know, but it was a lot of money. So as part of all this, we wanted to know, we from the Fish and Wildlife Service, wanted to know whether this was a good thing that we had done. Was it a worthwhile thing for the animal? And will the animals benefit from it? So prior to release, we wanted to be able to tag the animals so that we could follow them and evaluate their survival to the best of our ability. That was another political hoopla with the environmentalists from hell who didn't want us to do anything to their little otters because they'd already been traumatized enough. But we did get some compromises, and so minimally all sea otters that were released were flipper tagged, and then a subset of animals, and I believe it was 45 total ended up, had radio transmitters implanted in their abdomens. Unlike other species of marine mammals where you frequently hear about radio tag collars and things on their dorsal fins and all, it doesn't work on the sea otter, they found out over the years. It needs to be implanted. It's a free-floating object that goes inside a midline incision, and I'll show you in a moment. But anyways, 45 animals were selected, some of them here at the Valdez Center and some down in Seward, or at the Jackaloff facility. I'd like to show you just a few pictures, which I found, it was a fascinating event, again, to try to figure out how to do something in some fairly unusual circumstances. These animals were outside in the middle of Valdez Harbor, so we couldn't really very well pack up a bunch of animals and bring them over to the local clinic. So instead, some of these great Alaskan guys built us a lovely surgical suite on this floating dock out next to the floating net pens where the animals were kept. We brought all of the equipment out, and a team of people. For those of you who know Tom Williams from the Monterey Bay Aquarium, is the person who has basically worked out these techniques while working on animals in California, and he spearheaded this effort. In addition to doing the surgeries on these animals, other information was collected. Teeth were collected for aging. Measurements were taken. We tried to gather as much information to characterize the individual animal as possible. In addition to the radio transmitter, these little barcode dealy guys, I've forgotten the real name for them, were implanted just subcutaneously in the groin area, and they will stay with the animal or the carcass forever so that they can be read digitally to give a specific identification to the animal. This is a picture of the transmitter that actually goes inside the abdomen. It's about the size of a pack of cigarettes, and it's a non-reactive silicone-type material. One of the things that I found fascinating was we go through all this stuff with this animal that absolutely cannot have anything done to disrupt the insulation provided by its hair coat, or it will die. Well, then how do you do surgery? You know, where I came from, you always do this real nice clip job and you scrub and everything. But anyways, again, Tom had figured this out. They scrub it. They scrub all of the hair very nicely and cleanly, and then they put KY jelly on it and that's a comb. Take a comb and part it right down the midline, paste it down, and then put a plastic drape on it. Worked beautifully. And it's a very simple procedure. You just open it up, pop it in, and sew it shut. For this surgery, also another thing that was important was to be able to, as my boss told me, don't let any of these animals die. It was a very important thing to preserve the animals, but also from the public relations standpoint, it was brutal. What was done with these animals for anesthesia was we used an injectable narcotic and then used an antagonist to reverse it, and literally within 1 to 2 minutes, the animals were back in the water. We had a complete success, and it was a fascinating experience. So then we packed everything back up in the boat and went on and went down to do the rest of the surgeries down in Little Jackaloff Bay. And anyways, it was finally now fall, and it was time to release the animals according to the release strategy. So the animals were loaded back into their dog carriers, loaded onto big helicopters provided to us by the Coast Guard, and they were flown over into the pristine areas of the eastern part of Prince William Sound that had never been oiled. And then it was finally time. It was the day for the release. You can see this little guy in here, and he's just happy as he can be, and it was beautiful. This is, for those of you who are interested in, this is the little electronic gizmo, the wand, and the digital readout machine that reads those barcodes. This is used, I understand, in many species of animals now, and this is where you'd see the digital readout. So it's finally time to let them go, and it was great. I'll have to admit, for as depressing as the summer had been, it was finally truly wonderful to, at least for a few animals, to open the door and see them head out and catch their buddies, and they knew exactly what to do. And while they were all frolicking around out there, having a marvelous time back in the natural environment, the Fish and Wildlife Service biologists were heading to work. You can see this boat here is outfitted with the radio antenna for tracking. But in addition to just tracking these animals, a number of studies was conducted to follow the success of these animals, to monitor for reproductive success, for any kinds of other disease processes that may be going on, a whole host of things. So they went to work. So in conclusion, I'd like to say that it was a tremendous experience. I think we did a lot of very good things, but I think that this type of work, and especially the science that comes out of this, is very crucial, because I think we need to use this type of science to hopefully be able to be about the only ammunition we may ever be able to have to improve our laws or our regulations, to improve the safety. Another spill or another accident will happen, that's guaranteed. Not sure you can all see exactly what this is, but we've been seeing it on TV the last couple of days. These are the tanks of the Valdez terminal. This is the dock here that we've been seeing on TV. And this is the resident population of sea otters that live right in Valdez Harbor. I'd like to see this kind of science continue, and I'd like to see it really put to use, because I've got one more story I'd like to tell you. And this is about this big old guy that was brought to me in the fall. But the story behind him is one that absolutely just about broke my heart at the end of the year. He was a big, strong male, and he was found down in Windy Bay early in June. And he was fine. He wasn't oiled. There was no oil even in Windy Bay yet. But they knew the oil was coming towards Windy Bay, and so they thought to preempt any exposure, they would capture this animal, which they did, and they brought him into the Seward Otter facility, where he swam around and ate all that stuff and made new friends and had a marvelous summer. And he maintained a very good health status. At the end of the summer, he was loaded into that helicopter along with the rest and flown over to the eastern side of Prince William Sound. But he is a big dominant male, and as soon as he was released, he hit the deck and he swam right straight across Prince William Sound back over to Windy Bay. Windy Bay, in the meantime, had also been termed environmentally stable. It was completely clean, it was stable, and everybody had gone home. When he got over there, they found him 48 hours after his release, and they brought him into me. He was as oiled and as tarred as any animal I've ever saw early in the spill. So what that really brought home to me was that this spill happened in the spring, when the weather was absolutely as good as it gets in Alaska. We had all summer to work on it. Unfortunately for Exxon, but fortunately for the country, it happened to probably one of our wealthiest corporations who were willing to put the money and the effort into doing, I think, everything humanly possible to clean up the spill. But the bottom line is that when you do something this devastating in the marine environment, you just, no matter how hard you work or how much money you're willing to spend, you can't change it and you can't fix it. Prince William Sound isn't going to go away.